Okay, welcome back, everybody. We're ready to continue on in chapter 12. As I said at the end of the preceding Zoom lecture, instead of doing 3 lectures for chapter 12, I've decided to split it up into 4 lectures. So this will be number 3 of 4. So let me begin by sharing the screen as I always do and talk about drug resistance and how that develops in bacterial populations. And I think the one thing that we need to be mindful of is that the ability of a population of bacteria to resist the action of a drug like an antibiotic stems from the fact that it has the genetic wherewithal to negate the action of the drug. And there are a number of different ways that bacteria do that, and we'll be talking about those in just a few moments. But this particular ability to negate the action of the drug, again stemming from the genes present in the genome of the bacterium, held in the chromosome I should say, and or the plasmids of the cell. What we've discovered is that even before we expose a population of bacteria to a given drug, that there may be natural resistance already present in that population even before they've been exposed to the drug. And this has to do with the genetic viability and diversity that exists within most all populations, assuming that they're large enough. And so that's what this second bullet kind of talks a little bit about. And I think the last sentence sort of sums it up really well when we say, and I'll just quote it, some traits, drug-resistant traits we're talking about, are, quote, lying in wait, unquote, for an opportunity to be expressed and confer this adaptability on the species. In other words, protect it from the effect of the drug. I think this cartoon is kind of appropriate for the topic we're talking about here. So I'll let you kind of look that over and see if you can figure out what this bacterium is providing in the form of this piece of DNA. Can you figure out exactly what information is held in here that presumably this guy is going to receive? Yeah. Take a look and ponder that cartoon for a couple minutes. So how do populations acquire drug resistance? Well, there's 2 major mechanisms. And the first one here, which you note is the most common cause of drug resistance, relates to this notion that there are constant spontaneous mutations occurring within all bacterial populations, within the cells of those populations. Now, these may very well not be beneficial. Some of these mutations, as we've learned, some mutations may not have any significant impact on the population, but once in a blue moon, spontaneous mutations can lead to drug resistance by providing that cell, if it were exposed to the drug, the ability to negate the action of that drug. And if that cell survives, it's going to reproduce, and those daughter cells, if you will have the same genetics as the parent cell, i.e. having that ability to negate the action of the drug. So that would be beneficial to that population. You would have more individuals that would have the unique genetic blueprint, if you will, to negate the action of that particular drug. And that stemmed from a mutation that occurred in some previous cell. And that was then passed on as that cell divided. and those cells divided, and those cells divided, and so on and so forth. So let's, and we'll show you an example of this in a diagram in just a minute, but let's talk next about the second mechanism of how bacterial populations acquire resistance to drugs. And this stems from mechanisms that we've already talked about. This notion that cells can take up naked DNA from their surroundings. We talked about that back in chapter 10, I believe it was. Actually, it was chapter 9. Yeah, the end of chapter 9. Remember transformation and Griffith's experiment? Frederick Griffith. So cells can take up free DNA in their environment. And if that particular segment of DNA that came from that heat-killed cell, or that dead cell, however it was killed, has the instruction booklet to negate the action of the drug, and that becomes incorporated into the genome of the cell that took that naked DNA up, then obviously that cell is going to benefit. This is the same idea of the heat-killed S strain strep, remember, being picked up by the living R strain strep, and then the living R strain became S strain because they could produce this outer capsule, which increased its pathogenicity. It's exactly what we're talking about here. Transformation is one way that cells can acquire free DNA from their surroundings. The other mechanism we talked about in that chapter was the transfer of a plasmid. In this case, we call it an R factor plasmid, R standing for drug resistance, not to be confused with F factor plasmids, which allow the cell the ability to produce the sex pilus, right, which is needed for conjugation. So we're assuming that conjugation pilus has already been formed. Here we're transferring drug resistance plasmids, okay, across the sex pilus. And so this cell that had that plasmid presumably was drug resistant. This cell initially was not, but now it acquires that R factor plasmid and can negate the action of the drug. And then we also talked, remember, about transduction, this unique process that involved the use of bacteriophages to pick up DNA from an initial invaded cell and transfer that into a recipient cell. And that gene turned out to be beneficial because in that gene was the instruction booklet that allowed the cell to negate the action of, again, the drug. So these are the two primary ways in which drug resistance is acquired by bacterial populations, through this new gene transfer and spontaneous generations that can occur within the cell. So let's talk about ways in which microorganisms do indeed negate the action of the drug. How do they become resistant? How do they fight off the effect of the drug? There's a whole slug of different ways that cells can do this. One way, as you see here, is through the inactivation of a particular drug. And we'll use the example of penicillin. This should look somewhat familiar to you. We talked about it in earlier segments of this chapter. And we also talked about how some bacterial populations can prevent the activation or the effectiveness of this naturally produced antibiotic by producing enzymes. In this case, the enzyme penicillinase. Again, we know it's an enzyme because it ends in ASE. And what penicillinase does is it inactivates penicillin by breaking an important bond here that would ordinarily exist in the active form of the antibiotic. So if you can inactivate penicillin by affecting its chemistry, you have nothing to worry about as a microbial population. And as I said earlier in the chapter, there are some bacteria out there that can negate the action of this antibiotic that's being secreted by other bacteria in an effort to kill them. So this is a naturally produced enzyme. We also know that one way that drugs work on cells is by, first of all, docking with special protein receptors on the surface of the microorganism. Okay, so let's say this is drug X, and obviously under the normal situation, as I said, it would bind with specific protein receptors and be allowed into the cell, and then it would work on the cell by killing it in some way or affecting the DNA or whatever the case may be. Well, what some bacterial populations have evolved is the means to alter the receptor on the surface of the cell. Now, can these same drugs get in? The answer, of course, is no. Why not? Because they don't fit into the receptor site. We've altered it. Not we, but the cell, as a result of a mutation or as a result of acquiring the genes from another cell, like we talked about in the earlier slide, now reconstructs, if you will, or slightly modifies the existing normal receptor to the point at which the chemical, the drug, doesn't fit into it and therefore can't be allowed into the cell. Isn't that interesting? This third scenario talks about drug pumps, which are special proteins that some bacteria have. And so, as we've said, the normal entry for many drugs is to dock with a certain receptor and be brought into the cell. Okay, let's say it's brought in. Well, what does this mechanism involve? It involves taking this inactive drug pump and activating it to the point at which now it will pump the antibiotic or the drug out of the cell. So the bacterium activates a particular mechanism. It's called efflux. And it employs the use of special membrane pumps to pump that substrate or that compound. I shouldn't call it substrate, it's not an enzymatic reaction, but we're pumping that compound, that chemical, that drug out of the cell. Isn't that crazy? It can't cause problems if it can't stay in the cell. We talked a lot in the earlier lecture of this chapter about how some antibiotics influence translation of the protein by screwing up the ability of the two subunits from attaching, from translocation, right? Some drugs prevent translocation of the ribosome. There's a whole slug of different ways, right, that antibiotics target translation. Well, what can the bacterial population do? Well, what it can do is it can modify the binding site that the antibiotic would ordinarily fit into. Okay, so here's the antibiotic depicted by the green triangle. It ordinarily would fit in here, let's say, and prevent the interaction of the two subunits. Well, what the bacterium does is it says, I am going to modify my large subunit here in such a way that even though there may be antibiotic present, it cannot fit into that ribosome like it ordinarily would. Let's modify it so that it can fit in, and let's allow those two subunits to combine, and let's continue on with translation of the protein. So there's no harm done, no harm done to the cell, right? It's just negated the action of that antibiotic by modifying its protein structure. Basically, this is primarily protein and some RNA. And then finally, a fifth mechanism that your book talks a little bit about, and that is circumventing normal metabolic pathways. You recall one of those ways in which antibiotics affect drugs is by screwing up the metabolism. Remember our sulfonamide and trimethoprim antibiotics. Go back and look at that section. It talks about PABA and interfering with or competing with PABA. That's what the sulfonamide drugs do, or sulfur drugs. Well, here, what's going on is the bacterium evolves basically a workaround, right? So let's say this is trimethoprim or sulfonamides, which prevents, remember, the conversion of a given substrate into a product. And as you know from A&P and biology, metabolic pathways involve numerous enzymes working together to convert eventually these different chemicals into some desired end product that the cell needs. Well, obviously what's going on here is we go around the action of the drug. Yeah, the drug can stop this particular reaction, but instead of producing C from B, we're going to produce a C1, and then we'll convert C1 into D, and we'll convert D1 into a product. We just do a workaround. Fascinating, isn't it? Here's a hyperlink that I want you all to watch that reviews all of these different mechanisms of drug resistance. I think you'll find it to be a really, really excellent video. It lasts, I think, about 4 or 5 minutes. So make sure you watch that. So let's talk a little bit more about this idea of how the process occurs out in the real world. Now, if you've taken biology, which I know you all have in high school at least, you've heard of the phrase or the two words natural selection. And perhaps you associate that with evolution because that's often the mechanism we talk about in trying to explain evolutionary processes. We need to now think about evolutionary processes, and natural selection at the microbial population level. Okay? And you heard me mention just a few moments ago at the beginning of today's lecture that many bacterial populations, when you look at their genome or their gene pool, which are all the genes of all the cells within that population, that there are often these plasmids or perhaps mutations that occurred within the chromosome that may not initially provide any benefit to the population until it's exposed to the drug. Okay, so these sort of sit and wait, which was the terminology I think I mentioned when I gave that quote. So if we take a population like we have in this Petri plate, so all of these different dots are depicting cells. They might really better be thought of as colonies, but we'll pretend they're cells. And we've got diversity within that population. We've got some black cells and we've got some tan cells. Obviously, you see labeled here, those that are tan have the ability to negate the action of the drug we're going to expose to that population in just a moment, while these other guys do not possess the means to negate the action of the drug. And then we expose this population to drug X. Okay. Let's say again, this is a population of bacteria that's causing an infection in a person. We give that person an antibiotic. What's going to start to happen? Well, we're going to target a certain percentage of that population, those that cannot negate the action of the drug. They're susceptible to the drug. So you can see, of course, here the black cells are dying off, while those that have the wherewithal to survive because they negate the action of the drug, they survive and they can reproduce. If we were to continue to expose that population to that same drug, we would continue to kill off, the non-resistant forms of the cells, but there would be a lot more that, of course, would grow. Those that survived are not going to be affected by that drug because, again, they can negate the action of the drug in whatever way. So this builds up resistance in a population, doesn't it? You can't use that same drug. It's not going to have the same effect. You might kill some outliers that somehow survived that initial set of exposures, let's say, or pills. Let's say you're taking antibiotics. But a point will come potentially when the infection is not controllable by that particular drug because of the drug resistance that existed in that population prior to exposure to the drug. And so when we talk about exposing populations, bacterial populations to drugs, we are really talking about putting what we call selection pressure on that population. So the exposure to the drug, exposure of the population to the drug, is what allows those that are more fit, evolutionarily fit, in other words, they are able to survive, these gray guys, they're the more fit cells, as opposed to these that are less fit and will succumb to the action of that drug. The overall effect is to have more fit cells in that population as time goes on, as we continue to expose that population to that drug. And that's really the way in which drug resistance typically forms in a population over time. So what can we do to limit drug resistance? Well, there's a number of things we can do. And that's what this table kind of talks about. Physicians have to understand the infection. They have to make the proper diagnosis for the infection, and they have to prescribe the right drug for that infection. If they don't do that, they're certainly not helping the patient get over the infection, and they might inadvertently be increasing drug resistance in the long run. When we are prescribed an antibiotic to take, and it says to take 1 pill every day for 5 days or 10 days, we should follow those instructions. We should follow the instructions. If we have 10 pills, we should not stop after day 5 because our sore throat went away, and put those pills in the cabinet thinking, well, instead of going to the doctor the next time I have a sore throat, I've got 5 pills I can take, and I'll save myself the hassle and the money of having to go and get a new script. People do that all the time. You should not do that, because if you do that, you are increasing the risk of drug resistance. You are allowing for or selecting for increased mutations to prove beneficial to that population. You're trying to kill those cells off, not allowing any survivors. This third bullet really gets at the concept of synergy, which we talked about earlier in this chapter. Sometimes the combination of 2 or more drugs in concert with one another increases the chance of dealing with that infection, killing those cells. Taking one drug might end up not only increasing the time it takes to get over the infection, but you might actually also be increasing drug resistance to these other forms of bacteria. So sometimes a combination of drugs can be more effective than a single one. As we go to develop drugs, pharmaceutical companies are often eyeing the idea of using shorter term, higher dose antimicrobials versus longer term, lesser dose antibiotics. The notion of trying to minimize side effects is always there. We'll talk about side effects in just a couple of minutes, but extensive research, as you know, has to be done before any particular drug becomes marketable. It takes years and years and years usually for that to happen. And also trying to work around these mechanisms that these bacteria are always coming up with, evolving new techniques to negate the action of these drugs. We're trying to stay a step ahead of them, when really we're always a step behind them. We're really often reactionary as opposed to being proactive, because we don't know what the bacteria is going to develop, what sort of technique or what sort of mechanism will they evolve to circumvent what we're trying to do to kill them. So it's a constant effort. We're always reacting to the effects of the bacteria. Long term, just education, as we said earlier, making sure that patients take the entire dosage for the correct time frame, and trying to teach the American public, or even at the high school or middle school level, really. It's really what we should be doing. We should also be encouraging physicians to, as we said earlier, make a proper diagnosis and not be maybe prescribing drugs that are going to please the family member because, my daughter's sick. I've been coming to the doctor's office a lot. The doctor oftentimes, well, not oftentimes, but sometimes what they'll do is they'll prescribe an antibiotic when they know it's viral. Why do they do that? Because an antibiotic isn't going to touch a virus. Well, when a parent who's lost several days of work and has no more time to take off is coming regularly to the doctor's office, the physician can sometimes feel pressured to give a drug to please the parent when the physician knows that in another 2 or 3 days, that viral infection is going to subside. The parent's going to think it subsided because of the pill, the antibiotic that he gave his son. It's kind of almost unethical to be even thinking that this happens, but it does. It does happen. And when you are giving drugs that aren't effective, you're increasing drug resistance characteristics. You're causing problems down the road is really what you're doing when you do that. Especially valuable antimicrobials should be restricted in their use to only 1 or 2 types of infections. Yeah, if we start subscribing all sorts of antibiotics for all sorts of infections, we often aren't doing ourselves any benefit because sometimes we may run out of drugs to use. There's so much drug resistance. If we can set a few drugs on the shelf, if you will, and use those only when needed, that might prove to be much more helpful. The addition of antimicrobials to animal feeds must be curtailed worldwide. Yeah, we give antibiotics to animals all the time because we want to keep them healthy so that we can have them put on weight faster and get to market, and then we can get more money. But what we're doing is we're increasing drug resistance in bacteria and other pathogens in cows and pigs and chickens and all these animals that we eat. And there's also some studies that suggest that even in the meat that we eat, it's so laden with antibiotics. What effect is it having on our health? Yeah, it might have kept the chicken alive, kept it healthy till it was time to butcher it. But what long-term impact is that having on our health? I don't think it's probably good. Government programs making more effective therapy available to low-income populations. Yeah, one could even expand this to worldwide usage of drugs in countries where there isn't, the healthcare system in place. Again, not overuse, but targeted use in those populations that are susceptible really would be a good thing to do. And finally, the 5th bullet, they're talking about vaccines should be used whenever possible to provide alternative protection. Yeah, we can use drugs to cure disease, and we do that effectively. But if we can provide a vaccine and allow the individual to build up natural acquired immunity, that's beneficial in the long run because we're not being so reliant on using those drugs. We're letting natural immunity build up within the population. Okay, let's talk about some interactions that can occur between you and a drug that you might take. And it's always worthwhile noting that whenever drugs are taken by people, there will be a certain percentage of individuals that have a reaction to that drug. You will never develop a drug. Well, I shouldn't say never, but rarely will you develop a drug where you don't have to be expecting some small percent of the population having an adverse reaction to the drug. And that's why it's so critical to do all these tests. In the news just recently, yesterday's news, I think, there's concern about the AstraZeneca, Oxford AstraZeneca vaccine, I think it was, and even some concerns with the Johnson & Johnson vaccine, that there are some individuals who are having some adverse side effects, some blood clots and so forth. Now, I know that's a vaccine, but we can make the same argument for antibiotics and other sorts of drugs too. And that's why it takes so darn long to do all these tests, these years and years of testing to try to lower that number of individuals that suffer side effects. But again, it's almost impossible to eliminate any and all possible side effects. So one big concern, of course, is the effect that the drug might have on different organ systems. And your book talks quite a bit about this, specifically targeting the liver and the kidney as being two organs that often bear the brunt of chemotherapy. And I'll let you kind of look that over and read that over. The brain can also be impacted by different kinds of antibiotics and antimicrobials. I simply give this example because it's something that's very easy to see in young kids who have been given high dosages of tetracycline, which we talked about earlier in the chapter. And I did mention in that one slide that one of the side effects was the decoloration of the tooth enamel. Now, that might not sound like a life-threatening issue, and it's really obviously not for the most part. But as I mentioned in the red box, you would not want to give someone who's pregnant tetracycline, because we know that this is an example of an antibiotic or an antimicrobial that can cross the placental barrier, and the tetracycline can actually cause harm to the developing fetus's teeth and the developing bones. So one has to be always conscious of toxicity impacts, and different antibiotics have increased risk on different sorts of organs in the body. So make sure you look that over. Another major side effect that infects some people are allergic reactions to antimicrobials. And this particular photograph is showing some skin irritation. That is not uncommon. Skin rashes can occur quite routinely in certain individuals when they're exposed to certain types of antibiotics. I think of sulfa drugs. Some people are allergic to sulfa. And so these can cause, as we see here, some skin irritation, some rashes. Diarrhea is not uncommon because you're affecting the microbiota of the digestive system. And so if you know you're allergic to certain antibiotics, you of course tell your physician that, and we don't prescribe you those particular drugs. But sometimes you don't know that until you're a kid and you take a pill, take an antibiotic, and you have this allergic reaction to it. So we still steer clear of those. Disruption of the normal biota. In one of the next chapters, we're going to be talking about microorganisms that inhabit different parts of our body. And the gastrointestinal tract is a good example of an organ system that has a lot of different bacteria in it, all the way from the stomach through to the colon, really, and rectum. So when we disrupt the normal balance within normal microbial natural populations, we can have side effects. They're called super infections. So let's talk a little bit about how this works. Let's pretend you have strep throat. You go to the doctors, they do the throat swab, and they get the results back later that afternoon. And sure enough, you indeed have Streptococcus that's causing a lot of irritation. You're running a fever. There's some growths on your pharynx in the back, and the physician obviously then prescribes you an antibiotic to try to knock out the Streptococcus bacteria that's causing the strep throat. You take that antibiotic, it gets into the bloodstream, it gets taken all over the place. Of course, it's going to target the Streptococcus bacteria in your throat, and hopefully eventually after a couple of days you'll get over that, right? The sore throat will go away, the fever will abate, and so on and so forth. But let's look what happens to the bacteria in your intestine, in the small intestine. Now, prior to taking the pill, the antibiotic, we have a whole host of bacteria, different species, hundreds of different species of bacteria actually in our gut. And we're going to just look at two. We're going to look at the normal bacteria. We'll depict those guys as the pink triangles. And then we'll also depict the fact that within a normal microbial population, we also have potential pathogenic bacteria, denoted by the blue squares, that, as it indicates here, are normally held in check by the normal flora, the normal bacterial population, which certainly outnumbers the potential pathogens significantly, to the point at which the pathogens can't cause problems. Okay? That's the normal scenario. Lots of normal bacteria keeping the pathogens that are also living there, but in low numbers. They're kept in check, not causing any gastrointestinal upset. Then you take the drug. The drug is targeting, we hope, we assume, the Streptococcus in the throat, trying to get rid of strep throat. But what does it do to the natural, normal flora in the gut? Well, as you can see here, a lot of natural occurring cells, beneficial cells, we'll call them, they die from the effect of the drug. What survives are these pathogens. Guess what happens to the pathogens in a very short period of time? Well, they are no longer held in check, are they? Because these guys are killed. And so suddenly now we see an intestinal infection potentially set up, where the pathogens numbers skyrocket. There's nothing holding them back from really reproducing. And while our sore throat may be gone, or certainly symptoms abating, now we have what's called a superinfection that's developed in the small intestine. So this is just one example of how superinfections, whether it's here in the small intestine or a yeast infection in a woman, is not uncommon, of course, in the vagina, because when you take certain antibiotics, you knock out pathogens. Well, let's put it this way. You take an antibiotic that knocks out the yeast infection, or tries to knock out the yeast infection, but in essence, what it oftentimes can do is knock out the beneficial bacteria and allow those pathogens, in this case the yeast cells, to start to grow in very substantial numbers, just like we have here, and suddenly we have this issue. So anytime one takes antibiotics, yeast infections can often arise because they're not held in check anymore. Could have been from a previous infection of some sort, like we saw here with the strep throat. And the after effect, the superinfection, now becomes the yeast infection. This table kind of talks a bit more about different microbial, antimicrobial drugs. And we see here the antibacterials, antifungals, antiprotozoan, anti-helminth worm, antivirals. So we've talked about quite a bit of these, haven't we, in this chapter? And this is, again, talking specifically about how can these drugs have toxic impacts within the body. And while I'm not expecting you to memorize this table, I think it's worth noting again what are some common abnormalities, if you will, that can occur when you take certain types of antimicrobials. These are sort of side effects, aren't they? And they range from skin allergies to diarrhea, to decoloration of teeth, as we saw with the tetracycline, to damage to white blood cell and red blood cell precursors in the red bone marrow. Even some heart arrhythmias can occur with certain types of antibiotics. Hemolysis, that's of course rupture of the red blood cells. So as you look through this, damage to liver cells, some of these are not insignificant. Immunosuppression can knock down your immune system. Now, this is AZT. We talked about that with regard to treatment of HIV. So when you are prescribing drugs to patients, physicians need to have an understanding of what is the infection and what kind of drug can I give. And if that drug doesn't work, what's my plan B or plan C? And then we suddenly need to be aware of, are there any downsides or side effects that we need to be conscious of? And are those side effects more harmful to the patient than the bacterial infection potentially could be? So there's lots of things to consider. There's lots of balls in the air that the physician is juggling as he or she kind of goes to decide what to do when they're dealing with a patient with an infection. Okay, I'm going to stop this third lecture here, and we'll pick up and finish up the chapter with this next section, which I believe is section 12.6.